Okay, so this is a discussion about the micrococci. Particularly, we will be focusing on Staphylococcus and the Streptococcus. Okay, so under the family of Micrococci, it includes four genera: the Planococcus, Micrococcus, Tomatococcus, and the Staphylococcus. Okay, so first, the one that is actually medically important is the Staphylococcus. Okay, so these are actually a part of the normal flora on surface of primates as well as mammals, and thus including humans. Okay, so among these four genera, we will focus on the Staphylococcus. So, under the family of Micrococci, Stomatococci are part of normal flora, oral flora, and are now emerging pathogen in immunocompromised patients. So, what do we mean by immunocompromised patients? So, these patients are have a weakened immune system. Okay? So, mababa yung kanilang immune system. So it's not mababa but weakened. That's the proper term. So there's a disturbance in their immune system. So it's a possibility that there are other diseases, okay? Or like for example, they are suffering from AIDS already, okay? Or those who are undergoing chemotherapy, okay? So micrococci become pathogens when they are accidentally introduced into a susceptible host. Okay, so when they are introduced accidentally. So propensity to cause disease, it's the staphylococci that is medically important. Okay, that's why our discussion for this one will focus on staphylococci. Okay, so it has have long been recognized as important pathogens, and the most commonly isolated pathogenic species in order of pathogenicity is, of course, unang-una dyan is the Staphylococcus aureus. Okay, next we have yung mga coagulase negative Staphylococcus epidermidis, and then the Staphylococcus saprophyticus. So there are also other medically important Staphylococcus, namely the Staphylococcus lugdunensis and the Staphylococcus hemolyticus. Okay, but among them, the most common here is the Staphylococcus aureus and then the epidermidis. Okay, so if you hear the word Staphylococcus aureus, what disease association could you make? So we have there yung ano, what the toxic shock syndrome. Okay, associated with tampon use. What else? Also, it can also cause bacteremia, sepsis. Okay, what else? Pneumonia. So, it's because the Staphylococcus aureus is a part of the normal flora. Okay, so when we say normal flora, that is a bacteria that usually resides in the body, okay? Usually resides in or on humans that does not usually cause infection. Okay, so Staphylococcus is a normal flora. Okay, so morphology and general characteristics, of course, these are gram-positive bacteria. However, it was noted here that it may become gram variable with age. That's why when performing a gram stain, it should be from fresh culture, okay, or after a sample has been submitted, that is immediately. Okay, so do not perform gram staining from 48-year-old colonies, mga ganyan, 48 or 72 years old, because it may result to gram-negative reaction. Okay, so may occur singly, in pairs, or in tetrads. Okay, so they have varying they have varying, what do you call this one, morphologies under the microscope. So do not expect, kasi diba guys, we already mentioned a lot of times that for Staphylococcus aureus, these are in grape-like clusters. So it means kumpulan yung mga cocci. However, it's not all throughout for a sample. So there are cases wherein there are tetrads there, or singly, or in pairs, or in chain, and then there are those in cluster. So usually if that is gram, what do you call this one, if that is Staphylococcus aureus, majority of the microscopic appearance are in clusters, but there will be in singly, or in pairs, or in chain. So that is normal. Okay? So these tetrads, okay, kasi apat yan, okay, that is common or usual for the micrococci. And for staphylococci, usually in clusters. Okay, so that is from Greek term staphyle, which means bunches of grapes. That's why in some books they regard its microscopic appearance as grape-like clusters. Okay, grape-like clusters. So we have here division and pattern determines the arrangement. So we have here Diplococcus. Okay, so two cocci together. Pag Streptococcus, usually in chains. So it means, guys, for these types of morphologies, their division is in single plane only. Parang pa-side lang yung kanilang division. But for tetrad and the Sarcenia, their division is in 2 or 3 planes. Okay? For tetrad, that is in 2 plane, pa-X, okay, or pa-Y. Okay? X and Y axis. But for the Sarcenia, parang it has a 3D arrangement. Aside from dividing on the left and on the down part or the upper part, it can also divide on the other side. Okay, that's why it forms a three-dimensional, what do you call this one? It has a three-dimensional like structures. And for Staphylococcus naman, okay, yun din in more than 3 planes. Okay, that's why in any plane, so usually nagkukumpulan na sila, and then nag-overlap. There is overlapping, producing a characteristic grape-like cluster of cells for Staphylococcus aureus. So again, may I remind you, it was also noted here, rarely will a sample be composed of just one arrangement. The more complex the arrangement, the more likely scattered examples of simpler arrangement will be found. Okay? So, micrococcus, again, these are catalase positive, but they are coagulase negative. But for Staphylococcus, these are also catalase positive, but could either be coagulase positive or negative. Okay? So, later on, we discuss on how to differentiate Micrococcus from Staphylococcus. So, we have here the tetrads. I hope you can see this one. Okay? Clearly. So, yung parang apat in a square-like fashion. So, that is in tetrad, and that is more common in Micrococcus as compared to Staphylococcus. Although, you can see this type of morphology also in the microscopy, or pattern or arrangement in the microscope with some Staphylococcus species. So, it will grow on most laboratory media. Okay? So, it can grow on BAP, cap. Will it grow in MAC? For if we are talking about Staphylococcus and Micrococcus, will they grow in MAC? The usual MAC that we have, of course, not, because that is selective only for gram-negative. Okay, so for Staphylococcus, they may have smooth circular colonies with a buttery consistency as they grow. Okay, so butter. So it's like yellow in color. So Staphylococcus aureus has a golden or yellow pigmentation, or creamy or white pigmentation, just like here. Okay, so golden yellow. Okay, golden or yellow pigmentation. But for S. epidermidis, it's usually white. Okay, usually white. And for Micrococcus luteus, colonies with bright yellow pigmentation. Okay, Staphylococci are non-motile. These are usual for gram-positive cocci. They are non-motile. Okay, Staphylococci are non-motile. They are non-spore forming and aerobic or facultative anaerobe. Except for what? We have an exemption to that. The obligate anaerobe Staphylococcus species is the Staphylococcus saccharolyticus. Okay, so they are facultative anaerobic or aerobic except for Staphylococcus saprophyticus. Okay, so colonies are produced after 18 to 24 hours of incubation wherein they will appear as cream-colored, golden yellow, or white. Okay, sometimes referred also as buttery looking or buttery consistency. Okay, so there are some strains of Staphylococci which are fastidious. What do you mean again by fastidious? They require they have special requirements for growth. Okay, requiring carbon dioxide, hemine, and others. So we have here colonies of Staphylococcus aureus, creamy yellow in color. These are Staphylococcus epidermidis. Okay, white color. And we have here yung micrococcus also. It has yellow pigmentation. Okay, so both micrococcus and luteus and staphylococcus aureus may produce yellow pigmentation in their colonies. Okay, so we have here, if you look at this image, what do you notice? The first thing is, of course, the hemolysis pattern. Okay, that is beta hemolysis. So on BAP or BA or blood agar, the Staphylococcus aureus produce beta hemolysis. Okay, other Staphylococcus species produce alpha or gamma hemolysis. That's why when you are examining, so after putting your samples in BAP, on MAC, or if it requires CAF, so if you saw this type of colony wherein it has a large zone of beta hemolysis, can you already identify that that is Staphylococcus? Not yet, okay? So can you, after looking at that beta hemolytic pattern, can you already say that that is Staphylococcus aureus? Not also, you need additional testing. And for the definitive diagnosis for Staphylococcus aureus, you must perform up until to the coagulase test, up to the tube coagulase. Okay? So, that is for Staphylococcus aureus. So, I don't know if it has something to do with light here, but as you can see, what I can see here is this is not yellow colonies, but they produce a wide zone of beta hemolysis in the back. What if this same type of colony is also seen in MAC? What can you infer from that data? Well, if that's the case, it means that the bacteria is gram negative. So for Staphylococcus, it will only grow in the BAP and not in the MAC because there are bile salts and crystal violet that is inhibitory to gram positive bacteria present in the MAC media. Okay? So it was labeled here that this is Staphylococcus aureus. However, if you saw this type of colony, you cannot identify that this you cannot definitely identify it as Staphylococcus already. So if this is shown and if it was asked for you to identify this, so you cannot identify it. Okay, because the hemolytic pattern is not definitive. Okay, it is not definitive for any type of bacterial strain. So you have to do further testing. Okay, the phenylethyl alcohol inhibits gram-negative bacteria. It's like, for me to remember this one, it's like the counterpart of the MAC. Okay, so the PIA is a counterpart part of the MAC, MAC only allows gram-negative while PIA allows only gram-positive. Therefore, since this is selective media for gram-positive, Staphylococcus and Micrococcus can go here but not in MAC. On the other hand, for the Enterobacteriaceae, which are gram-negative, they can grow in MAC but not in PIA. Okay? So, biochemical identification, Stomatococcus versus all others in the family. So, Stomatococci are catalase negative. Micrococci and Staphylococci are catalase positive. That's why we cannot use yung catalase test to differentiate between the genus Micrococcus and Staphylococcus because both of them are catalase positive. Okay? So, take note of that. Okay? So, for stomatococci, these are catalase negative. However, these are what do you call this one? These are non-medically important. Okay? Or, um, yes, they are not usually associated with human infections. Okay? So, for the catalase test, we already discussed this one. So, what do we usually utilize as the reagent here? That is hydrogen peroxide, 3% for aerobic or facultative anaerobe. Pero kapag obligate anaerobe, that is 15% potassium hydroxide. If you remember yung discussion natin sa bacillus, right? So that is again bubble production. So among these two, okay, the first one that is catalase positive, this is negative, this is positive, and this is negative. Okay, so enterococcus, these are under the family also of the streptococcosae, okay, or streptococcus. Although they were formerly kasi they were under group D, Streptococcus, but then again they were reclassified into a different genus that is the Enterococcus. But these are catalase negative just like Streptococcus. So that is Staphylococcus. Okay, can we again, guys, take note in this example, what they utilize as the testing organism is the Staphylococcus aureus. But it doesn't mean that if I show you this type of picture, okay, and I ask you what type of organism is that, the answer is that uh, it is an unidentifiable. Okay, you cannot identify it using this test alone because this is not a definitive testing. Okay, so even if I show you this one, for example, this is letter A and this is letter B, identify the organism that was utilized for this test, wherein it is positive. Okay, so you cannot definitely say that, okay, immediately that is Staphylococcus aureus. What you can say is that it could be Staphylococcus or Micrococcus species because these are positive for catalase test. Okay, something like that. Okay, just in case lang naman. So, bacitracin. So, in the bacitracin test, here we utilize 0.04 or 0.04 units of bacitracin. And what I usually think about this one is bacitracin is for micrococcus. Okay? So, kasi the positive reaction, di ba, is sensitive, not the resistant. So, that's the usual case. Kapag positive means sensitive. So, here, for me, bacitracin is for micrococcus. Okay? So, you can use this one to differentiate. Kasi, diba, we cannot use catalase test to differentiate to differentiate micrococci from staphylococci. However, we can use other tests. Like, for example, the bacitracin. We can use the bacitracin to determine if that is micrococci or staphylococci. Both. Kasi if it shows that it's catalase positive. Okay, so definitely you cannot say immediately that the gram-positive cocci, which is catalase positive, is always staphylococci. So to differentiate that, you may use the bacitracin. So bacitracin, okay, so if that is sensitive or that is positive for bacitracin, that is under micrococci or micrococcus. Pero kapag resistant siya, staphylococci. Okay, modified oxidase test. Okay, also this one is for micrococci, which is micrococci that is positive for this one. Okay, so if you have to remember also what do you mean by positive and negative. what is the minimum inhibitory concentration or the zone of inhibition for micrococci. I think that is greater than 10. So check if I'm right. Let's try to remember. Yeah, I think it's 10, greater than 10. Okay? For the micrococci, the microdase or the modified oxidase test naman, that is blue, blue to blue purple. Okay, so that is positive. Okay, so that is for micrococci, bacitracin, and the modified oxidase test is for micrococci. I think it that way. Okay, para maalala ko na si micrococci yung sensitive and si micrococci din yung positive for the modified oxidase or the microdase. So you can use that one to differentiate yung catalase positive, gram-positive cocci, which is also catalase positive. Okay, so you may use bacitracin or the modified oxidase test to rule out if that is Staphylococcus or Micrococcus. Okay, so family Micrococcaceae, lysostaphin, that is a protease that breaks the glycine peptide linkage in the cell wall of Staphylococcus species. Lysostaphin, on the other hand, is positive for what? Staphylococcus. So that is positive for Staphylococcus. So we already have. So let's review again. Bacitracin, sensitive for micrococci. The other one, the microdase, modified oxidase, that is blue or positive for micrococci. Lysostaphin, on the other hand, this is positive for staphylococci. Okay, iba na. Yung first two, that is micrococci, lysostaphin, that is for staphylococci. So we have here, okay, without looking at this one, so tago muna natin. Without looking at the description of the image, we have bacitracin susceptibility. This one, this is resistant because as you can see, there's no zone of inhibition, right? There's no zone of inhibition. Unlike here, there is, okay? As you can see, there is a zone of inhibition. So, this one, the first one, or the upper portion, is it Staphylococcus or Micrococcus? This is Staphylococcus. Okay? Kasi nga that is resistant. Okay? So, if you have two testing organism, bacitracin, in bacitracin susceptibility. And this one, this is the micrococci or the micrococcus. Kasi nga that is susceptible. Okay? How about this one? If for example, that is the reading up to, pang ilan? 7 or 8, that is resistant. Kasi diba, greater than 10 ang sensitive. So, you measure from this part up to this part. So, that's greater than 10. Definitely, this is greater than 10. Because usually, the size of this disc is 6 to 7 millimeter. Konti na lang. Okay? So, 4 na lang. And that would already mean that is susceptible. Okay? So, konti na lang yung hahabulin. Okay? For staple, which means bunches of grapes. The coccoid means berries. Okay? The coccoid means berries. And the staple means that is bunches of grapes. Okay, so this one, that should be Y. Staple, bunch of grapes. And the coccoid, berries, means berries. Okay, so again, paulit-ulit, these are gram-positive cocci that are catalase producing. So, we review na. Bacitracin, negative. Microdase, in bacitracin, it is resistant. Under microdase, that is negative. And in lysostaphin, sensitive or positive. These are facultatively anaerobic bacteria except what? We already mentioned this one, which is an obligate anaerobe, meaning as in it grows only in the absence of oxygen. That is, if you can remember, that is, again, Staphylococcus saccharolyticus. Okay, Staphylococcus, that is S. paralyticus. Okay? So, the Staphylococci are non-motile, non-spore-forming. They are non-spore-forming unlike yung gram-positive bacilli and yung mga Clostridium, which are spore former. And this is a glucose fermenter. Okay? So, they produce acid or they ferment glucose. So, these are normal inhabitants of the skin. Therefore, they are normal flora. Okay? Normal flora of skin, mucous membranes, and intestine. That's why there are cases of bacteremia. Okay? So, bacteremia associated with staphylococcus, endocarditis as well, even pneumonia. Okay? And yun nga, some staphylococcus also lies the genitourinary tract of females. That's why staphylococcus can also cause UTI. Okay? So, and aside from that, sometimes also the staphylococcus may cause yung mga acne or pimple formation, yung super mga acne type. Okay, may mga pus formation, di ba pag nagka-clog yan, or sometimes there is bacterial infection. Okay, because we had cases before na they aspirated the pimple, okay, of a patient who has a lot of acne in her face and in the gram stain, that is gram positive cocci, and then since the request was culture and sensitivity, we proceed with the sensitivity testing, and yun nga, identification and sensitivity, it was identified that was Staphylococcus aureus. Okay, so these are spherical, meaning pabilog, circular, that appear in clusters, singly, tetrads, okay, or in chains. Okay? But usually, pag mga chains, yeah, yung mga coagulase negative staphylococcus na yun. Okay? So, culture, we don't need mga special requirements because, they can, they can grow in back. Okay? Colonies, 4 to 8 millimeter, appear creamy. For me, guys, you don't need naman to memorize yung how big or how small yung diameter of the colonies because sometimes it varies eh. There are cases wherein that is Staphylococcus pero yung diameter niya is 3 or 2 millimeter and we do not measure that one in the lab. We do not report or we do not measure, okay, yung diameter of the colonies. That's why I'm not very particular with this, yung mga numbers. Okay? Creamy, white, or light gold, buttery looking. Beta-hemolytic pattern that is very usual for Staphylococcus aureus. Other species that are not Staphylococcus aureus may have alpha or they could be non-hemolytic. And according to Mayhon pala is that we have the alpha beta, diba? And then the alpha prime. Okay, alpha prime, and the other one is that is non-hemolytic. So, parang it was mentioned by Mihon that there should be no gamma hemolysis because there's no hemolysis at all. So, it should be termed that instead of using gamma hemolysis, it should be termed as non-hemolytic or non-hemolysis. Okay? So, Staphylococcus arrangement, this one, as you can see here, we have the grape-like clusters, this. So, it's very obvious that the Staphylococcus aureus will not all of the arrangement that you can see under the microscope is in bunch of grapes. So, there are singly, there are the cocci, diplococci, okay, there is in chain, but majority, okay, or most of the arrangement is in what? Okay, in clusters. Of course, do not count yan na, ay, mas madami yung paisa-isa. No, of course, kung ano yung mas prominent, mas nakikita. Okay? So, yung Staphylococcus aureus is also regarded as a common opportunistic pathogen. Okay? Kasi nga, since this is a normal flora, kasi when we say normal flora, it resides in the body and usually, it will not cause an infection. Okay? So, Why is it that it is termed as opportunistic? Kasi the term opportunistic means that the pathogen usually does not cause disease in healthy individual. Okay? Pero it can be pathogenic if a person is immunocompromised or unhealthy already. She or he has a weakened immune system. That's why, yun nga, si Staphylococcus aureus is also regarded as what? As opportunistic pathogen. So, we have here staph in tissue. So, yun yung mga gram-positive cocci. So, this is under the gram staining. So, this is a tissue sample. So, that is also intracellular already. What we mean by intracellular? The bacteria is within or located inside a cell. So, here, that is inside the cell that is inside a um I cannot identify if this is a polymorphonuclear, so most likely this is neutrophil that is inside a neutrophil. Kasi 'di ba? Why is it, ma'am? Why is it inside the neutrophil? Kasi neutrophils, they have the capability to phagocytize. So parang kakainin nila. So they will eat the bacteria when these are present in the tissues or even in the blood, because these are our defense system. Okay, so they will phagocytize it. Okay? So, if we differentiate Staphylococcus aureus from coagulase negative Staphylococcus aureus, so actually, I already mentioned it, yun na, we use the coagulase test to differentiate it among the different species of the Staphylococcus aureus. Okay? So, in the presence of coagulase, fibrinogen is converted into fibrin. That's why our reagent here is the rabbit plasma. Kasi the rabbit plasma, of course, contains fibrinogen. So, what do you call this one? The coagulase uh, that is present in the colony or in the bacterial cell wall can lead to a reaction with the fibrinogen. So, magkakaroon siya ng reaction with the fibrinogen, i-convert niya into fibrin, that will then result to the formation of clumping, okay, or coagulation that is visible to the eye. So, yun na yung magiging positive test result. Okay? So, all other Staphylococcus species are collectively called as coagulase negative Staphylococcus ascons, like sila epidermidis, si Saprophyticus. Okay? But guys, take note, yun nga, kasi not only Staphylococcus aureus is positive for coagulase test, but we will discuss that later. But usually kasi, if that is from human sample, from human isolate, if that is coagulase positive in the tube method, we report that already as Staphylococcus aureus. Okay? Pero, depende kasi yan sa protocol ng lab. They may proceed up to the identification if that is coagulase negative, but they may also report that as a coagulase negative Staphylococcus aureus. So after testing it under the tube coagulase test, depending on the protocol of the laboratory, pwedeng hindi na nila i-identify kung anong type ng coagulase negative. Pwede na nilang i-report na, okay, so this is not Staphylococcus aureus, so we will report it as coagulase negative Staphylococcus. Yun na yung magiging reporting. Okay? So, magdedepende yan sa protocol, magdedepende din yan sa training in different countries. Okay? Yung mga standard na siniset ng different countries. But if that's already negative for tube coagulase, that is regarded as cons. So, kasama na dyan yung epidermidis, intermedius, saprophyticus, okay? And others. So, there are two different coagulase tests. So, we already discussed this one, yung slide and yung tube. Pag slide, it reacts with the, what? The fibrinogen reacts with the bound coagulase present in the cell wall. Pag tube coagulase naman, that is, the fibrinogen reacts with free or extracellular coagulase that leads to clumping or fibrin clot formation. Yun na yung nagka-clamp, nagko-coagulate. So, actually, that is fibrin clot formation. Okay, mannitol fermentation. Okay? Sa mannitol fermentation, I hope I have an image of that one here. Wait, we'll discuss it first. Okay, so in the mannitol fermentation, we already discussed this one in culture media discussion. So, sa mannitol fermentation, Staphylococcus aureus and some Staphylococcus saprophyticus are positive. So, what would be the positive test result in the mannitol salt agar? So, that is what it will appear as yellow colonies because of the utilization of mannitol, thus producing acid, turning the phenyl red indicator to yellow. Pero kapag epidermidis, negative siya. So, kung anong color niya, pinkish pa rin. Okay? Like the color nung agar or nung indicator which is phenol red. Sa Denase naman, so Denase is, Staphylococcus aureus is positive for the Denase. So, that is also an agar. Pero si epidermidis and saprophyticus, they are negative. So, ano na naman yung positive sa Denase? That is parang what? Disappearance or clearing, di ba? Clearing around the colony. So, para siyang beta hemolysis. Pero sa Denise agar, which is green. So, we have actually a picture here. So, this one, ito yung sinasabi kong positive for mannitol. Mannitol fermentation. This is Staphylococcus aureus. Okay? Um, Staphylococcus aureus. So, you can use this one to differentiate, but it's usually kasi in testing, the tube method is enough to identify it. The tube coagulase test is already enough to identify it as Staphylococcus aureus. Pero kapag nagdududa ka pa rin because there are other Staphylococcus which are positive, then you may proceed with using the Denase test agar and the mannitol salt agar. So, mannitol salt agar, positive Staphylococcus aureus. Denase test, eto nagkakaroon ng clearing around the colony. So, eto stricto. Strict of bacteria, which is Staphylococcus aureus kasi nga positive siya. Okay? So, eto negative, see? Between the two, sino ba yung magpo-positive? Staphylococcus aureus. Sa Staphylococcus epidermidis will have a negative test result. Okay? Can you identify a colony or a bacteria that is negative in the mannitol salt as Staphylococcus epidermidis? Hindi. Kasi all Staphylococcus that are not aureus will have this type of colony here. Magne-negative sila. Ganon din yung principle sa Denase test agar. So, lahat ng Staphylococcus species, magne-negative sila. So, hindi mo pwedeng gamitin yung negative test result as a definitive testing. Okay? So, yung positive lang. So, differentiation with cons. So, yun na. You may use this one. Okay? So, differentiation. We usually only distinguish between the cons and urinary tract isolate. Kapag sa urinary tract. So, kapag usually mga wound infection, mga bacteremia, or others, pwede mo na siyang i-report as coagulase negative. Except pag urine yung sample. Okay? Kapag urine yung sample, you have to identify if that is epidermidis or saprophyticus. Okay? So, to differentiate between, dito kasi, pag urine yung sample mo, you saw an alpha hemolysis or gamma hemolysis, coagulase negative. In the urine sample, you have to establish if that is epidermidis or saprophyticus. To do that one, you can use, or we may use novobiocin using 5 microgram disc. If that is sensitive with greater than 17 millimeter zone of inhibition, that is epidermidis. So, this can be definitive already or pwede na tong definitive na to. So, kung naging ganyan yung resulta, nag-zone of inhibition na ganyan, pwede mo na siyang report. Okay? So, that is pag sensitive greater than 17. Do not forget yung bacitracin greater than 10. Novobiocin greater than 17. So, that is epidermidis positive and saprophyticus resistant siya sa novobiocin. Okay? So, Para tandaan yan, sino ba yung, di ba, novobiocin, meron siyang dalawang eye. Sino ba yung mas maraming eye? Si epidermidis. So, siya yung sensitive. Okay? So, novobiocin, dalawang eye for sensitive. Mas maraming eye si epidermidis. So, siya yung sensitive. Greater than 17 or equal to 17. Okay? So, resistant between the two, without looking at the description, this one, ano to? Saprophyticus or epidermidis? Saprophyticus. Kasi nga resistant siya, wala siyang zone of inhibition. Eto, kung umabot pa ng 17, that is epidermidis. Okay? So, other way to identify as causative agents, um, under serologic testing, ito under serology, usually, pag serology kasi, what you measured here are antibodies. Okay, so rising antibody, pwede din namang page typing. Okay, so yun yung gagamitin mo. But usually, pag mga ganitong way of testing, these are utilized for epidemiologic investigation. Even the epidemiologic investigation. Pag may mga outbreaks, mga ganyan. Or may mga studies na ginagawa. Hindi for patients. Okay? Na for treatment. Okay? Mga study. So, toxins. Toxins produced by Staphylococcus aureus. So, we will just go over this one kasi this one should be under the lecture discussion, hindi sa lab. But anyway, it's here already. So, let's just discuss it. Hemolysins. Sa Staphylococcus aureus, so this is actually a part of the virulence factor ng Staphylococcus. Okay? What's that? Okay, so remember, yung si Staphylococcus aureus is beta hemolytic, pero it is beta hemolytic, but it may produce four different type of hemolysins. Okay, so that is alpha, beta, gamma, and the delta. Alpha toxin in particular may produce extensive tissue damage. Beta hemolysin, that is known as the hot cold lysine. Okay, bakit siya hot cold? Kasi na-enhance yung hemolysis niya if in-incubate mo siya sa incubator 37 and then ilalagay mo siya sa freezer. So, na-enhance yung hemolysis. So, another is yung coagulase wherein we already discussed this one. Okay, so ano ba yung action ni coagulase? Since, di ba in vitro, when we are performing this in the laboratory, it forms coagulation or clumping. So that is actually one of its way to avoid yung host immune system in vivo. Kapag nasa loob siya ng katawan, pwede niyang mai-avoid ang phagocytosis. Okay? Paano? So yun nga, it will coagulate. Okay? Magpo-form siya ng coagulation so that it can protect itself against the neutrophil. So pag nag-coagulate na siya, okay? So, mas malaki na siyang particle. It is within the fibrin plug. Hindi na siya mapaphagocytize, hindi na siya makakain ng neutrophil. And yun nga, kapag masyadong mataas yung coagulation because of the coagulase produced by Staphylococcus aureus, that can lead to intravascular coagulation, lalong-lalo na sa lungs. Okay? So, those are possible complications. So, dalawa, free or bound, free or extracellular coagulase combines with serum component to produce a thrombin-like activity. Okay, to cleave that fibrinogen, yun nga, para magkaroon siya ng fibrin clot formation. So, yung bound coagulase binds to fibrinogen and cell surfaces so that it convert it to fibrin. So, ganun din, leading to coagulation or agglutination. Okay? What do you mean by ubiquitous? guys. It's a very usual term in what do you call this one in medical field, especially in what do you call this one in bacteriology. When you say ubiquitous, that is found everywhere, usually seen anywhere, in the soil, in the air, ganyan. Kahit saan. Kumbaga, kahit saan pwede mo siyang makita. Okay? Existing everywhere at the same time. Parang ganun yung meaning ng ubiquitous. So, if that is ubiquitous in nature, parang it means na pwede mo siyang makita kahit saan. Okay? Kahit saang part ng katawan. Parang ganun. Okay? So, kahit saang environment. Staphylococcus are ubiquitous and found as normal flora. So, it means they are part of the normal flora, or they are usually found as part of the skin, or they are usually in the skin, sa mucous membrane. So, nandyan na sila normally. Okay? So, there are CNS strains, usually Staphylococcus epidermidis. So, these are part of the normal flora of the skin. Kasi that they can also cause, yun nga, infection of the CNS. Okay? So, CNS, so, what else? Staphylococcus aureus is part of the normal flora of the nasopharynx in 10 to 40% of the population. So, kumbaga, it's normally there. It's just, it's part of, ano na, kumbaga, it's part of the human species that the Staphylococcus aureus is just normally a normal flora of the humans, but not all. Okay, not all has this one. So they are opportunistic, so or facultative pathogens. Even yung Staphylococcus aureus can also be opportunistic pathogen. Okay. Okay, so another, we already mentioned and discussed some of this, like si bacitracin, yun nga lang ang gagamitin mo dito na culture media is pwedeng ba, pwedeng molar hinton. So, again, positive for micrococci. Dito naman kabaliktaran. Okay, furazolidone susceptibility. So, ang positive naman dito, ang susceptible is si Staphylococci, greater than 15 millimeter zone of inhibition. Okay? Sa lysostafin naman, para siyang lysostafin. Pero si lysostafin, ang zone of inhibition niya para maging positive, that is 17. Diba? If you remember. So, sa furazolidone susceptibility test, gagamit ng 100 microgram of furazolidone sa bath. Lysostafin, okay? So, hindi pare-pareho yung, ano natin, tawag dito? What do you call this one? References, di ba yung kanina is 17, dito naman 10 to 16, zone of inhibition. Okay? So, greater than 10, siguro, greater than 10 is accepted. So, lysostafin for Staphylococci, modified oxidase for, eto, parang what I can remember is 5 minutes yan, dun kay Baileys. Diba? That is, this one is positive for micrococci, blue color or blue purple. So, ang reagent niya contains tetramethyl P phenylenediamine in dimethyl sulfoxide. So, bacitracin, micrococci, furazolidone. Bacitracin 10, furazolidone, that is for Staphylococci greater than 15, lysostafin greater than 10 for Staphylococci. So, furazolidone and lysostafin for Staphylococci, modified oxidase for micrococci, growth on furoxon twin 80 oil red o agar, micrococci. So, nakatatlo na tayo for micrococci. Bacitracin, microdase, growth on furoxon twin 80 or oil red o agar. acid production from glycerol, staphylococci. Okay? So, 3/3 na pareho ng 3. Okay, acid production from glycerol, staphylococci. So, we can rearrange this and the oxidation fermentation reaction, that is staphylococci. Okay? So, pwede nating i-rearrange. Bacitracin, modified oxidase. Growth, that is micrococci. Ito naman, what is that again? Furosolidone, positive for staphylococci. Lysostapine, positive for staphylococci. Acid production from glycerol with erythromycin, positive for staphylococci. And then see oxidation and fermentation, that is staphylococci. Okay? So, fermenter si staphylococci. Okay? So, dito sa oxidative fermentative, oxidative si micrococci. Ito, it does not ferment glucose. Si staphylococci naman is fermentative. Okay? So, nag-change yung color into yellow because of acid production. Okay, so Staphylococcus aureus through coagulase positive, and that is the most virulent. Okay, so citrate utilizing organisms may yield false positive result. Kaya ang gagamitin dito na, what do you call this one? Na false positive kapag citrate yung ginamit, usually it comes from blood bank, diba, pag mga human sample. Kaya nga hindi siya recommended. Aside from that, that is also infectious. It may also be infectious. Kaya rabbit plasma yung preferred. And the anticoagulant must be in EDTA kasi nga citrate may lead to false positive. So, staphylococcal species are initially differentiated sa coagulase test. Okay? However, not only Staphylococcus aureus is positive, pwede din sa coagulase. Positive din sa tube method si Intermedius, Delphini, Lutre, and Hicus. Okay? So, that is, ilagay na lang natin siya dito. Also positive for coagulase. However, kasi itong mga to, so why is it not that if it comes from human sample, if that is positive for tube coagulase, we already reported as Staphylococcus aureus. Kasi itong mga to, these are from animals, and these are not common sa humans naman, sa human infection. So that is the reason why. Another is that si Lugdunensis, okay, Lugdunensis and Sclaferi is positive sa slide coagulase test naman. Itong dalawang to. Kaya, because they contain also the clumping factor, yung bound coagulase in their cell wall. So, sa culture Bob, they have golden yellow color and are beta hemolytic. So, this one, we are referring to the Staphylococcus aureus. So, they are, what do you call this one? They can halophilic, okay? They can tolerate a high amount of salt. Kaya nga, it can be cultivated by adding 7.5 to 10% in ECL if you want the media to be more selective. So again, these are also halophilic organisms. So, paulit-ulit to, reagent, rabbit plasma, anticoagulant, EDTA. Okay, slide method, yun nga, presence of the bound or the clumping factor, positive result pag may clot. That is within 10 seconds. Pag tube naman, reading, that is after. Sensitive but definitive. Okay, so it means if it is positive from human sample and if it is positive in the tube method, we reported already as Staphylococcus aureus. So, ang minimeasure naman dito is the extracellular or free coagulase. Okay, free coagulase or the Staphy coagulase. So, the Staphy coagulase or the free coagulase kasi, it is a thrombin-like molecule. Okay, since it is a thrombin-like molecule, which is also called coagulase reacting factor. So, to form coagulase CRF complex. So, the coagulase CRF complex, it resembles yung thrombin. Okay? Since it resembles thrombin, it can indirectly, okay, it can indirectly convert fibrinogen to fibrin, leading to clot formation or coagulation. And usually the clot is formed in 1 to 4 hours of incubation in the, what you call this one, in the incubator. So sa incubator mo ilalagay yung tube method and read it after 4 hours. If no clot appears, the tube should be left at room temp. Okay, at room temp and check the following day. So kapag wala daw, walang clot, hindi mo siya i-report immediately as coagulase negative staphylococcus. Instead, you will take the tube out of the incubator and you place it in the room temperature and you check that after 20 hours. Kasi di ba tapos mo nang i-check yan for 4 hours. So, the next 20 hours. So, parang 1 day or 24 hours from the time na ginawa mo yung test. Okay? So, you will assess that again after 20 hours the next day. Okay? So, fibrinolysin activity is enhanced at 37 degrees Celsius kasi. Ano ba kasi yung fibrinolysin? So, why is it that some Staphylococcus aureus has a negative test result after 4 hours? It's because some Staphylococcus aureus species produces fibrinolysin. And this fibrinolysin activity is enhanced in the incubator. So kapag na-enhance siya, yung na-form na clot will will be dissolved because of the presence of the fibrinolysin. Okay? So, kaya nga siya ilalabas from the incubator. Kasi nga, na-enhance yung fibrinolysin effect sa incubator. Okay? So, ilalabas mo siya into room temperature. Okay? So, kapag negative pa rin siya after 20 hours, okay, aside from the 4 hours, that can be reported already as coagulase negative staph. Okay? So, yan yun. Si Staphylocoagulase, yan yung term sa ibang book, or that is known as the free coagulase. So, yan yung ina-explain ko na it can form, okay, it reacts with the thrombin-like molecule, CCRF, so that will form into coagulase CRF complex. Si coagulase CRF complex, it mimics the thrombin. Si thrombin kasi, it activates the fibrinogen to become a fibrin clot. So, yun yung nangyayari. Since it mimics the thrombin, the coagulase CRF complex can activate the fibrinogen to form the fibrin, resulting to clot formation. Okay, so ito yung ina-explain ko kanina. The clot formed in the tube may undergo autolysis or autolysis because of the fibrinolysin, which is enhanced at 37 degrees Celsius. Okay, that's why you do not report immediately na coagulase negative siya after 4 hours of incubation. Kailangan mo muna siyang i-observe at room temp for another 20 hours. So, if no clot appears after 4 hours, it should be left at room temp for additional 20 hours of incubation. Pero kapag nag-positive na siya sa 4 hours, yun nga, pwede mo na siyang i-report actually as Staphylococcus aureus. Okay? So, other tube coagulase positive Staphylococci species, Hicus, Intermedius, Lutrei, Delphinis, Clefairy, and the subspecies coagulans. But usually kasi itong mga to, they are not found in human samples because these are not part of the normal flora ng human. Okay, instead sa animals sila. Okay, so kaya nga nakalagay dyan. Human usually pag si Staphylococcus aureus. Yung mga may asterisk, common in human or veterinary disease sa mga sakit associated with yung mga may kagat, animal bites, ganyan, exposure to animals, something like that. So, ito yung tube coagulase. So, again, that is rabbit plasma in EDTA at 37 degrees Celsius. Okay? Pag negative, ilalabas into room temperature another reading after 20 hours, excluding yung 4 hours. So, a total of 24 hours pa rin yun. So, eto na, mannitol fermentation. Na-discuss ko na to. Okay, so paulit-ulit, mannitol salt agar, mag-yellow si Staphylococcus aureus kasi nga it ferments mannitol, thus producing acid, turning the mannitol into yellow. Okay? So, meron din siyang high salt concentration, si mannitol salt agar. Okay, that's why it can be selective. So, si Staphylococcus aureus are facultative halophiles and thus they can grow up to 10% salt. Okay, so sa MSA, both si Staphylococcus and Epidermidis, they grow on this, pero si Staphylococcus aureus yung nagpo-ferment. So, siya yung nag-yellow. Epidermidis, hindi. Tellurite glycine agar, so jet black colonies. Under this one, jet black colonies of Staphylococcus aureus appear. And sa polymyxin sensitivity test, resistant dito si Staphylococcus. Okay, so negative. Polymyxin sensitivity, negative si Staphylococcus aureus. VP positive. Okay, VP positive naman siya. So, yun nga, Staphylococcus aureus positive. So that is when differentiating Staphylococcus aureus from Staphylococcus intermedius. Okay? Positive dito si aureus, negative si intermedius. Denase, we already discussed this one. Positive si Staphylococcus aureus. So, saan ito ilalagay? Siyempre sa incubator. Kasi pag mga ganyang media yung gamit, sa incubator talaga. Okay? Okay, so another strain, so this is also Staphylococcus aureus. However, it developed resistance to certain types of antibiotics. So we have the MRSA. So there are different types of MRSA, hospital-acquired MRSA, HA-MRSA. So, kumbaga, the resistance was acquired while you are staying in the hospital, okay? Or you were able to acquire this type of bacteria when you were in the hospital. Another is community-acquired MRSA, CAMRSA, and the last one that is the healthcare-associated community-onset, HACO-MRSA. Pero nandito din siya sa slide natin, so don't worry about that. Okay, so it is a type of Staphylococcus aureus strain that is resistant to antibiotics such as methicillin, napsilin, and deoxycycline. It can be acquired after a prolonged stay in the hospital, sa ICU and burn patients, close contacts with individuals who are carriers of the organism, after effects of a broad-spectrum antibiotic treatment, and exposure to nasal secretion. So yun nga, as I was telling you, you can acquire this one directly. The strain mismo, you can acquire the strain from other patients or other individuals in the community, or you may acquire it with prolonged treatment of antibiotics. Okay, kumbaga, the bacteria, the Staphylococcus that is being treated within your system acquires a resistance against methicillin. And when we say methicillin resistance, it does not only excuse me, it does not only refer to resistance against methicillin, but as well as other type of bacteria which are methicillin, like kagaya ni napsilin and C-oxacillin. Okay, sa chromogenic test, it changes in the color of MRSA colonies within 24 to 48 hours using chrome agar against colorless colonies of non-MRSA. So, under this chromogenic test, yung MRSA colonies nag-iiba yung kulay nila. Okay? So, hindi kagaya nung colony na hindi siya MRSA. Wala siyang resistance against methicillin and other related antibiotics. Colorless yung colonies niya. Okay? In the antimicrobial susceptibility, yun nga, 85% of Staphylococcus aureus na produce beta-lactamase to inactivate penicillin. That's why you can, in these cases, do not use yung mga penicillin type ng antibiotic. So, if that's the case, kapag penicillin resistant na yung strain, you may use yung methicillin. Okay? Kasi yun nga, because of, if a bacteria kasi produces beta-lactamase, they can inactivate, sisirain nila yung beta-lactam ring ng antibiotic na penicillin. So, hindi mo na yun pwedeng gamitin kasi magiging ineffective yung antibiotic. Ito na yung mga sinasabi nating antibiotic resistance. Okay? However, yun nga, meron na namang MRSA. So, pag MRSA na kasi, the resistance is due to the change in the cell wall that leads to altered binding of antibiotics. So, you cannot use cephalosporin, hindi rin pwedeng streptomycin, tetracycline, sulfonamides, methicillin, nafcillin, and oxacillin. So, if that's the case, ano na yung remedy mo na antibiotic? Vancomycin na. Okay? So, yan kasi, this is detected during the, what do you call this one? During the susceptibility testing. Iba yung culture and sensitivity sa culture, i-grow mo kung anong bacteria meron sa clinical sample. Sa susceptibility testing, kapag may nag-grow na bacteria of medical importance, i-check mo yung antibiotic na pwedeng gamitin, kung resistant pa ba siya or susceptible. Okay, so for the minimum inhibitory concentration, so when do we say that magkaiba kasi for Staphylococcus aureus using cefoxitin, di ba this is termed as methicillin resistant Staphylococcus aureus, MRSA, pero hindi kasi methicillin yung ginagamit na pang-test. Okay? So, it was changed to oxacillin in the 1990s, and nowadays, ang recommended by CLSI guidelines is cefoxitin. Okay? So, cefoxitin, yung test that is utilized to determine the methicillin resistance of Staphylococcus aureus. Okay? So, Staphylococcus aureus, cefoxitin. Okay? So, if the zone of inhibition for Staphylococcus aureus is less than or equal to 21 millimeter, that is considered as resistant. Okay? So, take note of that. Okay? That is for Staphylococcus aureus. Iba naman yung case kapag mga cons. Coagulase negative. Pag mga cons, coagulase negative Staphylococcus species. Okay? So, that is SAO, Staphylococcus aureus. Using cefoxitin pa rin, if the zone of inhibition is less than 24 millimeter, that is considered as resistant. And that is reported as methicillin resistant cons. Okay? And that one is methicillin-resistant Staphylococcus aureus. Pero pag greater than 22, so susceptible, hindi mo siya tatawagin MRSA. So kapag greater than 25 naman dito sa cons, zone of inhibition, hindi siya MR cons. Okay, MR cons or methicillin-resistant coagulase negative Staphylococcus aureus. Okay, so yun, MR cons, methicillin resistant, MRSA, and MR cons. Okay? Or wala yan, MR cons. So, in these cases, ang choice, ang treatment of choice, if that's the case, is already the vancomycin. Pero yun nga lang, may mga vancomycin resistant strain na naman. Okay? So, notes to remember, So the resistance of Staphylococci to penicillinase, resistant penicillin, is due to the altered penicillin binding protein known as the PBP2A or the PBP2 in the cell wall, which is encoded by the MECA gene. So the MECA gene, if a MECA gene is present in a Staphylococcus aureus, that codes for the resistance. Okay? So, it renders the oxacillin or methicillin or cifoxipine ineffective for treatment. So, hindi mo na pwedeng gamitin yung mga yun. So, kasama na dyan ang methicillin, oxacillin, napsilin, cloxacillin, and dicloxacillin. Pero ang ginagamit nga, remember, is the cifoxipine. Okay? Cifoxipine ang ginagamit. Nowadays, yun yung recommended to assess the methicillin-resistant status of Staphylococcus species. Okay? So, kapag meron silang MECA gene. So, si epidermidis naman, that is a normal flora also of the skin. That's why, yun nga, our skin is dirty. Technically, when it comes to presence of bacteria, these are actually dirty. Okay? So, it is a contaminant of medical instruments, catheters, yung mga indwelling and IV, intravenous, ganyan. That's why, yun nga, very important ang disinfection. CSF shunts, prosthetic heart valve implants, yung mga implanted medical devices. So, sa BAP, yun nga, non-hemolytic siya, white, opaque, small to medium sized pinheads. Hindi siya yellow. Okay? White lang siya. Biochemical test, yun nga, that is negative for coagulase, both tube and slide. Negative din siya for the mannitol salt agar. Okay. What else? Susceptible siya to novobiosine. Yun nga, yung differentiation nila ni Saprophyticus, di ba? So, we can differentiate this one kapag urine sample, novobiosine sensitive si Staphylococcus epidermidis, whereas si Saprophyticus is resistant. So, that is the community-acquired UTI in young sexually active female. Kasi nga, it resides or it lines in the epithelial cells that lines the urogenital area. Okay? So, sa urogenital area siya. So, in back, the colonies appear as white, opaque, slightly larger than pinheads, and are non-hemolytic, although some strains produce yellow. When you say pinhead kasi na colony, parang pinpoint lang. Pinpoint as in, parang tuldok-tuldok. Ganun yung pinhead. Okay? So, in urine culture, the presence, even if that is less than 10,000 colony forming unit, that is still considered as significant. So, magdedepende kasi yan sa laboratory protocol, sa SOP. May mga laboratory na kapag less than 20 yung colonies, kapag urine sample, hindi na nire-report. Okay? Pero kapag meron namang mga laboratory kapag greater than 10, nire-report na nila yun. They proceed also to anti-susceptibility testing. So, biochemical test, this is considered as coagulase negative Staphylococcus. Negative din siya for MSA. Okay, antimicrobial test, yun nga, resistant siya. That is resistant to the novobiosine. Hindi kagaya ni epidermidis na susceptible. On the other hand, si lugdunensis, that is cons by tube method. What do we mean by that? It is coagulase positive sa slide method. Pero negative siya sa, what do you call this one? Negative siya sa tube coagulase. It can be confused with SRUs kapag slide coagulase lang yung ginamit. This is more aggressive as compared to other cons when it comes to infectivity. It contains the MECA gene. Yun nga, that codes for oxacillin resistance, methicillin resistance also. So, some related infections include infective endocarditis, meningitis, septicemia, UTI, and skin and soft tissue infection. Okay? So, however, yun nga hindi common yung i-determine mo siya up to the species level. So, usually nire-report na siya as cons. So, novobiocin susceptible, epidermidis, capitis, hemolyticus, hominis, lugdunensis, saccharolyticus, and D. Warneri. Okay? Pag mga resistant naman sa prophyticus, coniae, closiae, and xylosus. However, kasi yun nga, tumato, these are not normal floras of the humans, instead sa animals sila. Itong mga to. Kaya these are not considered as medically important bacteria. Sa Hemolyticus, Staphylococcus saprophyticus, yun nga, it is commonly isolated cons. So, reported siya in wounds, bacteremia, endocarditis, and UTI. So, si Hemolyticus, just like the uh, aureus, is capable also of what? Of vancomycin resistant. So, BRSA, kapag vancomycin resistant Staphylococcus aureus, that is BRSA yung reporting. Okay? So, laboratory diagnosis. So, na-discuss na natin to. Oxidase for micrococci, growth on fructose micrococci, anaerobic acid production, staphylo, resistance to bacitracin micrococci, lysosome micrococci, pag lysostaphin, staphylococcus. So, we're done with this. Staphylococcus, beta hemolysis, non-hemolytic, grade white, staphylococcus, it may produce yellow pigment. Si epidermidis, non-hemolytic, and yung iba can be alpha hemolytic. Si saprophyticus naman, larger colonies. So, some produce yellow pigment, so nagkakaroon ng yellow coloration of colonies. Hemolyticus, medium size. Okay, they can be non-hemolytic or alpha hemolytic. And lugdunensis, often hemolytic. But then again, colony morphology, kaya for me, okay, this can give you an idea, but this is not definitive. Okay, so it means a lot of things. You cannot utilize yung colony morphology and microscopic examination, even hemolysis for definitive diagnosis. So you have to do further testing. Another is the PYR, the pyrrolidonyl aryl amylase activity. It can differentiate SRUs from other coagulase positive Staphylococcus. Okay? So, sila Lugdunensis intermedius and Scleiferi, which are positive for PYR. Okay? So, you can use that to differentiate it with Staphylococcus aureus, which is PYR negative. BP positive naman si Staphylococcus aureus. Staph aureus, VP positive, S intermedius, negative. So, another, VP lugdunensis, hemolyticus, Scleiferi, they are also VP positive. Kaya nga you cannot use this one to differentiate it with this kasi pare-pareho silang VP positive. So, to differentiate it with hemolyticus, lugdunensis, and Scleiferi, use the PYR activity. Okay? Clumping factor, so if they do not produce the clumping factor or the free coagulase or the staphycoagulase, that is termed as cons. Pero kapag urine isolate, kailangang i-identify kung saprophyticus or epidermidis. So, presumptive identification of saprophyticus by using novobiocin susceptibility. It should be resistant. Okay? It should be resistant. So, we have here identification of staphylococcal species. So, gram stain kapag gram positive. Okay. We perform catalase. Catalase, kapag negative, streptococcus yan. Kapag catalase positive, that is, pwedeng staphylococcus, pwede ding micrococcus. Kaya nga, you have to perform coagulase. Kapag nag-coagulase ka, that is, pag nag-positive, Staphylococcus aureus. Pero pag negative for coagulase, you may perform oxidase and bacitracin susceptibility. Positive for si oxidase, positive for oxidase, yung modified oxidase test. Positive for oxidase si Micrococcus, susceptible din siya sa bacitracin. So, kapag oxidase positive, bacitracin positive, or susceptible, Micrococcus yan. Pero kapag negative for oxidase test, negative for bacitracin, that is coagulase negative Staphylococcus aureus. Okay? So, kapag hindi yun yung sample, pwede mo na siyang i-report as cons. Coagulase negative Staphylococcus is PC. Pero kapag yun yung sample mo, you have to figure out if that is what? That is saprophyticus or other coagulase negative like epidermidis. Pero other also kasi are susceptible. So, ang nakalagay dyan is coagulase negative staphylococcus lang. So, pwede mong i-report as cons sa urine. Pero kapag naging resistant siya sa novobiocin, you report that as Staphylococcus saprophyticus. Okay? Rapid identification, ito na yung mga serologic testing, rapid agglutination test kits. So, meron silang plasma-coated carrier particle kagaya ng latex. So, if they detect the clumping factor and protein A, okay, so it will react. So, it is particularly useful in MRSA. Pwede din gamitin ang PCR, RT-PCR, real-time polymerase chain reaction. Okay? So, the PCR pala, PCR yun. Iba pala yung RT kasi pag RT is reverse transcriptase yun. So, this is PCR to determine methicillin-resistant and methicillin-sensitive Staphylococcus aureus. So, other methods is qualitative nucleic acid hybridization assay. Kapag mga prepared smears in blood culture, pwede din yung Malditov, which is a type of mass spectrometry. Okay, kasi nga requirement na daw ni FDA na pag-integrate ng automation. So, CLSI guidelines do not require routine antimicrobial susceptibility testing for S. saprophyticus from urine. Okay? Because isolates typically are sensitive to agents commonly used to treat UTI. So, kahit hindi ka na mag-proceed to what we call this one, susceptibility testing. So, yun nga, another is, because many Staphylococcus aureus produces this type of enzyme, penicillins are not recommended anymore to treat Staphylococcus. Kasi yung beta-lactamases, which is an enzyme, they inactivate or they break down yung beta-lactam ring na present sa penicillin. Okay, so kaya hindi siya ginagamit, yung mga penicillin. So, methicillin resistant, that is resistant to methicillin, napsilin, oxacillin, cephalosporin, di ba? Yung na-mention kanina. Merong methicillin resistant Staphylococcus aureus, may methicillin resistant Staphylococcus epidermidis, community-acquired methicillin resistant Staphylococcus aureus, hospital-acquired, community-acquired yung CA, HA, hospital-acquired, healthcare-associated community-onset, yung HACO. Okay? HACO MRSA. So, vancomycin remains the treatment of choice for MRSA. Before, oxacillin was generally used for the detection of methicillin resistant, pero yun nga, as I mentioned, yung bagong guideline ng CLSI, cefoxitin na yung dapat ginagamit. Okay? Cefoxitin na yung dapat ginagamit for the assessment of MRSA. What else? Latex agglutinations to detect these altered PDPs as well. So, they provide alternative method for testing and confirmation of oxacillin resistance. And yung gold standard for MRSA detection of the MEC gene is the PCR. Okay? PCR or the molecular nucleic acid probes. kapag McAG, gold standard PCR. Another here is we have this D test. Okay, D test. Kasi yung resulta niya dapat parang it's like D. Merong flattening of in the clindamycin zone, which is adjacent to erythromycin. So parang D-like pattern siya. So this one is an example of microlide resistance. So in the microlide resistance, this is used in the staphylococcal skin infection. Pag mga skin infection caused by staphylococcus. So additional testing using modified double diffusion disk or the D-zone test might be useful when discrepant macrolide test result are obtained kapag resistant si erythromycin pero si clindamycin is susceptible. Kasi dapat yan is pareho silang resulta. Pag resistant yung isa, dapat resistant yung isa. Sensitive yung isa, sensitive din dapat si clinda. Or kung sensitive or susceptible si erythromycin, susceptible din dapat si clindamycin. So, they go together. Okay, the result of these two antibiotic go together. Pero kapag yun nga, may discrepant, proceed with the D-zone test. So, yan yun, the D test positive isolate. Okay? So, nagkakaroon ng flattening on the side of the clindamycin. So, what does that mean? So, that means that is positive for microlide resistance. Okay? So, we have here the differentiation among the most clinically significant coagulase positive staphylococci. Kasi di ba I mentioned that it's not only Staphylococcus aureus is coagulase positive up to the tube method. Meron ding iba. Okay? So, yun nga lang, usually, pag mga ganyan, kasi siya naman yung beta hemolytic. So, i-compare mo kasi yung beta hemolytic siya, okay, and other testing. So, yan, you can use that one. As you can see, look at the, tube coagulase. Pwede silang mag-positive lahat. Okay? So, you can use other tests to establish that is Staphylococcus aureus. Pero yun nga, kapag nakita mo na siya sa lab that it caused a beta hemolytic pattern, big beta hemolytic pattern, you can establish na, okay? So, pwede na yon na correlation with the tube coagulase. Kasi, itong mga to naman, rarely or hindi nga sila nagkakos ng beta hemolytic pattern. Okay? Because they do not produce those types of hemolysins. Okay? But another is, if in case you want to establish, hindi mo na pwedeng gamitin yung navavirus resistance kasi it is negative. Yung PYR test, polymyxin B resistance, meron din na naman ulit, may variable. Okay, so sa PYR negative siya. Same with HICUS, pero you can use other tests to differentiate it with HICUS, like yung sa clumping factor. Negative siya for slide. And negative din siya sa acetone production. So, VP negative. Magbe-VP negative siya. Okay? So, that ends our discussion with Staphylococcus. Medyo mahaba kasi talaga ng Staphylococcus. Another discussion will be uploaded about Streptococcus naman. Thank you!